if anybody has anything. Everything seems clear? Good. Are you ready, Helena, already, although we are a bit ahead of time? So our first keynote is Associate Professor Helena Kilpinen. She is working at the University of Helsinki and High Life, human geneticist, combining molecular, experimental and computational approaches. And she has, before establishing the team in Finland, has done international career, so with postdocs in Geneva, the EMBL, EBI, and also as a group leader at UCL. And she will talk about visualizing complexity. So welcome, Helena. Haluatko käyttää tätä vai tota?
Okay, well, good morning, everyone. I can just introduce myself a little bit more while my slides load here. It's an honor to kick off this meeting. It feels very special for someone like me because I actually started off by studying biology, the kind of biology where you catch butterflies and sift through plankton, not far from here, actually. But then I met genes and I became a geneticist, and kind of slowly over the years, through my PhD and certainly during my time as a postdoc, I transitioned painfully, but eventually successfully, to becoming a computational biologist. And kind of being here today is really a testament that that path worked. And we are certainly avid users of various bioconductor tools. We've used tons of them over the years around different topic areas. And today what I'm going to talk to you about is some recent work we're doing in the group, specifically in the context of how we phenotype single cells. Okay, so my group is primarily interested in human neurodevelopment, and specifically how it can go wrong and how diseases can appear. And being a geneticist, we of course are very interested about mutations and genetic variants, what they do to our cells. In terms of neurodevelopment, there's a huge group of rare genetic disorders, neurodevelopmental disorders, which are individually super rare, even unique, but collectively affect up to 5% of children globally, so a kind of substantial group of disorders. And these tend to be intellectual disability syndromes with varying kinds of symptoms. All are generally caused by de novo loss of function mutations in different kinds of genes, but a common thread has in the recent years started appearing through large-scale genomic sequencing studies, which is that very often these mutations land and affect genes that are part of the epigenetic machinery of the cell. So this means that these factors that get partially damaged are those that run around in our cells and carry out things like methylation, histone post-translational modifications, and so on. So despite being individually rare, there seems to be a point of convergence here in the types of genes that are being affected. We also work a little bit with common variants, how common variants affect neurodevelopment, but the focus here today is going to be on these rare disorders. Now, we know these genes. We know which genes are mutated and affected, which cause disease, but we actually know very, very little how that happens. What are the cell types that get first affected? Where do the mutations cause their first damage that then eventually leads to this individual being born with a congenital neurodevelopmental disorder? So lots of questions. And kind of just trying to figure out the kind of earliest time point in which we can look at these effects in vitro. Now, why do we care about cellular phenotypes? Why don't we study something else? The fact is that if we know the genetic variants and the mutations that cause problems, the closer we are to that genetic variant, the better chance we have to identify phenotypes that might be directly affected by this variant. if we think of things like genome-wide association studies, which are of course the state of the art in common complex traits in humans at the moment, you need thousands, even millions of humans, carefully phenotyped humans, to be able to identify these kinds of genetic effects. Whereas in rare diseases, this is not, of course, possible. You don't have these kinds of sample sizes. You might even have an N of 1 person with a disease or a mutation. And there it helps if you're close to the mutation. You kind of look what the earliest bits in that chain is. And we know from already some time ago that when it comes to genetic variants affecting cellular phenotypes, they act in a fairly consistent way. They can affect things like transcription factor binding, which then goes ahead and affects chromatin accessibility, transcription, proteins, and so on. So these effects tend to be pretty consistent. Now, as a geneticist, it's been a fun 10 years or so. We've kind of learned to sequence everything, so we can find a lot of variants and mutations. That's no longer the bottleneck. Instead, when it comes to fields like mine, there are many bottlenecks, but these are kind of among the two most important ones. One is that access to disease-relevant cell types still remains limited. We often tend to study things that are easy to get. Often this means blood cell types, because blood is, of course, very easy to get from all of us. And the second is that we're still very focused on sequencing technology. As I mentioned, we can sequence everything. We can do wonderful things in the scope of functional genomics, sequence the transcriptomes, all kinds of things. But ultimately, sequencing readouts and things like transcriptomes is not the same thing as function. So we're still lacking a little bit tools, molecular tools, where we could go beyond things like transcripts, proteins, to the actual cell level phenotypes in a scalable way. So towards this first point, towards accessing disease-relevant cell types, just a brief notion of this. So my group has chosen to focus on human-induced pluripotent stem cell models. Can you raise your hand if you've never heard of these cells? Okay. They are actually just approaching their 20th anniversary. They were first discovered in mouse in 2006, and they got the Nobel Prize in 2012. These are basically, the technology means that you can take any cell from any one of us. It can be a blood cell, it can be a skin cell, and using a particular combination of transcription factors, you can push that cell backwards in time in a way. You can push it backwards to becoming a stem cell. And what this means is that it is a pluripotent stem cell, which you can then work with in the lab, in the dish, and differentiate it to any kind of cell our human bodies have. So this is a very powerful system. Before these cells came about, the way to do this was to use embryonic stem cells. Now, as all of you can imagine, there's a whole lot of ethical and other challenges when it comes to embryonic material. So this was really a game changer for the field, that you could really just take blood cells, make them into iPSCs, and then start differentiating them to different things. In our case, we use them to make neurons. So we use a number of different iPS differentiation systems in the group. They're all more or less focused on cortical cells, so the cortex of the brain. They vary in length and in style, but one of the central ones that we've used quite a bit here is one that goes up until about 70 days in vitro, starting from the iPSC, so the stem cells, and then progressing kind of over time through progenitor states to early neurons and then to more mature neurons. And they more or less express the kind of markers we would expect to see in these types of cells when you stain them as they go. The challenge with iPSC models is that they are very, very variable. There's donor variability, there's line-to-line variability, there's variability in the cell types you actually make in the dish. And therefore, one of the first things we wanted to do when we started working with this system was to characterize it, to really kind of see what are the cell types we're making in the dish when we use this system. So nothing about disease here, just healthy lines. If we put them through this system, what happens and what kind of cells do we make? So we turn to single-cell RNA sequencing for this. This is kind of the state-of-the-art method for the in vitro world, because it really is very powerful in exactly this, kind of telling you what you are profiling. So we collected data from these three time points here in this UMAP plot, which many of you may have seen before. It's a cluster plot where every dot is a cell clustered based on the transcriptomes. And this is now all three time points together. And in any kind of single cell experiment, probably the biggest challenge, or one of them, is how you annotate your cells. How do you add a label to each of the cells and call it an interneuron, call it a progenitor, call it a neuron? And there's many methods and tools out there to do this type of work. We here resorted to using an external reference. So we used basically single cell data derived from actual fetal brain to see and compare what we're making in the dish. Long story short, overall, we recapitulate what we would expect. We see progenitors progressing to early neurons, interneurons, and so on, to more mature neurons over time. So this all made sense to us. But one thing that's worth mentioning, if you look very carefully, you can see gray dots there somewhere in the kind of lower layer of that UMAP. And the gray is not labeled here, but the gray means that we had a lot of cells that we actually couldn't annotate in this way. About 60% of the cells from this experiment very nicely matched fetal cell types, but the rest, about 40%, did not. And we scratched our head a little bit like, okay, are these just some kind of off-target weird cells we're making in the dish? Well, some are, but actually many of them turned out to be present in other data sets, organoid data sets, brain organoid data sets, and so on. So in addition to matching fetal cell types, we certainly have a large fraction in the dish of cells that are specific to in vitro. Now, whether these are actually useful for us, we don't know. But it's good to remember that when making cells in vitro, in addition to making things that you probably want to make, you might make all kinds of other things too, which may interfere with your interpretations. So, okay, this was the transcriptomic space of this experiment. We now knew what we were making in the dish. But a key property of neurons in particular is that when neurons develop, they transition from being, broadly speaking, round cells to becoming the very complex cells that we know are neurons with all kinds of axons and projections and so on. And these kinds of morphological changes happen gradually over time as they develop, and also in a very kind of controlled and a temporally controlled manner as well. And of course, these types of processes are kind of prime candidates to be influenced by genetic variants. So we wanted to have something that we could measure this with, this morphological transition. And this brings me to cellular imaging, specifically cell painting. So we took these same cells from the same experiment and profiled them also using cell painting. Now, these are not measurements from the exact same cells. This technology isn't quite there yet. There is now some attempts to bring these two measurements to the same cell, but we didn't have that at the time. So these are from the same pool of cells, but in a way parallel measurements, just to point that out. If you've never heard of cell painting before, not to worry, most people haven't when I give this talk. Cell painting is basically a multiplexed high-content imaging-based assay intended to stain and profile the morphology of the cell and its organelles. The trick of this assay, compared to many other similar things, is that it uses fluorescent dyes rather than antibodies. And why this is important is that it makes it cheap. And this is, of course, desirable when we think about the second point in that bottleneck slide I had, where we think about scalability and being able to run things sort of over and over again. So the idea of this assay is that you have a combination of fluorescent dyes that stain different components of cells, organelles, the mitochondria, nuclei, and so on. And then you basically, the aim is that you kind of paint as much of the cell as possible to come up with a representative image of each cell. Imaging data, of course, is a whole field of its own. As you know, it's really rapidly expanding. Tools are being developed all the time. The methodologies to actually analyze these kinds of data are not as established as in something like transcriptomics. But effectively what we did here is that we tried to work with the data much the same way as we would with single-cell transcriptomic data. That means that from the individual images, we would segment individual cells, and then of course do a bunch of QC and things like that. But in the end, what that meant is that we had, similarly to the single-cell expression data, we had measurements of these imaging features from individual cells, with which you can then work the same way as you can with expression data. So this UMAP cluster plot here is actually of the images or the imaging data rather than expression. Now the feature space is much smaller. So this is here about 150 features. It can range up to 500, maybe even 1,000 features, sometimes depending on exactly what goes into your experiment. But it's single cell, but the feature space is smaller. And the difference here is that when you look at clusters like this here, we don't have the same kind of expectation as we do in the expression space, where we broadly speaking think that the clusters represent cell types or cell states. We don't have that expectation here because we don't necessarily expect the morphology of these cells to have anything to do with cell types. And this is, of course, good. This is what it should be because we're hoping to measure something different than what transcriptomics is giving us. But that also means that when clustering these data and when working with it, it's a bit harder because we don't necessarily know what the clusters represent. We here made an effort to look at the underlying images in the final clustering that we had to make sure that they seem to actually correspond to something real, which hopefully means that the clusters would also, biologically speaking, represent something real. But that's, of course, a kind of unknown in the imaging space, that the interpretability of these features is not as good. But once you have these data, you can then work with it much the same way as you can with gene expression. Here is, again, the UMAP on which we have projected a couple of example features. There's cell area on the bottom one, and then an example from the mitochondrial channel on the top. And of course, cell area is interpretable, you know what that means. But then when it comes to these other types of features representing the organelles or some other even more cryptic things, we don't necessarily know what it means. But of course, in our case, when you look at a UMAP like this, we actually know something about this UMAP. We had a time course experiment, so we know exactly which cell came from which time point. And that helps us put these features in the context of differentiation time. So we know that, for instance, this mitochondrial example that I'm showing you here, the kind of lowest feature values are there on the top, and then you kind of get the higher values towards the right there. And this, broadly speaking, corresponds with the time points. So we found that this mitochondrial feature and others, this is just an example, were broadly speaking the best markers of differentiation progress in this system. So this was almost a kind of nice validation for us that actually these phenotypes, these slightly cryptic imaging phenotypes that we are measuring do seem to capture something real in this system. Okay, so this has all been on healthy cells, just kind of characterizing the model, seeing what we could learn from the model. But of course, as I mentioned in the beginning, we were interested about what we could do in terms of understanding disease. And my disease example today comes from Kabuki syndrome. Kabuki syndrome is one of these genetically defined neurodevelopmental disorders that I mentioned in the beginning. It's a chromatin disorder, so it's the genes that are affected in this syndrome are chromatin modifiers. It's a rare disease, mostly characterized by intellectual disability and very kind of particular facial features if you've ever encountered this syndrome, hence the name. And we had iPSCs from a small number of Kabuki syndrome patients. So we put those cell lines along with a couple of extra wild-type cell lines through this very same differentiation protocol. We didn't take them all the way. We differentiated them only up until the first time point, so the day 20 time point. And then we collected cell painting data from these and put it together with the previous data set. So spend some time in figuring out how to best integrate these data sets together. Now, of course, with imaging data, when you have case control samples like here, you can do much the same things as you would with differential expression studies. You can look for differentially abundant features between these groups, which is basically what's shown there on the right. Now, these are not individual features that are being plotted. These are clusters of features. And for the sake of this talk, the labels don't really matter here. But one thing to point out is that you can see labels saying day 40 enriched in the Kabuki samples. Now, there is no day 40 cells here. We differentiated these cells only up until day 20. So this was curious. We were a bit puzzled what was going on here. You can similarly see that in the healthy cells, you have an enrichment of clusters of features labeled day 20. So there's an enrichment of day 20 features, as there should in the healthy cells, but for whatever reason, there's an enrichment of features that resembled, based on the previous experiment we had, that resembled day 40-like cells. So this raised our attention because, as it happens, one of the few cellular phenotypes that is consistently being reported in studying the cellular effects of these disorders is a kind of differentiation timing phenotype. So you have cells that are either committing to a neuronal fate faster than they should, so kind of racing ahead on the differentiation or the developmental path, or vice versa, that they take their sweet time to become neurons. So the timing of development and differentiation can be affected in these disorders. So this is why we really notice this. Now, this brings me back to the interpretability question. We, of course, now having observed this timing phenotype or potential timing phenotype, wanted to understand a bit more what was going on there. And this brings me back now to the expression data. So this is the expression UMAP you saw before. We wanted to basically go back to it and see if we could use the transcriptomic data that we had from the same cell types, so the same experiment, to see if we could add a layer of information to the imaging features. And to do this, we turned to a fairly basic linear prediction model. We basically tried predicting one modality from the other. You can look for genes whose expression is most predictive of certain image features, or of course vice versa. You can do both ways. And to our surprise, this actually worked reasonably well. We used the coefficients from this model to build links between expressed genes and image features. And what this allows you to do is to then basically bring all of the annotations you have in the expression space to the imaging space, which means that you can then start putting these image features in the context of cell types, genes, pathways, and so on. So now here, this is again the expression, or still the expression UMAP, but projected on it is this example image feature from the mitochondrial channel that I've used throughout. So we can see how the intensity of this particular channel is distributed in the gene expression space, so in different cell types and so on. And this is really quite powerful for us, because then that means that we can go back to the Kabuki setting, for instance, and start asking that, okay, these imaging clusters that we find overrepresented in the Kabuki samples, what are they? What genes do they link with? What kind of pathways do they affect? And this kind of opens up the toolbox of what you can do with gene set enrichment analyses and so on, also with the image features. And by doing so, this actually confirmed to us that those processes that were downregulated in the Kabuki samples had to do with the cell cycle, and vice versa, those that were upregulated in the Kabuki samples had to do with cellular differentiation. So it really did seem that by kind of backtracking to the expression data, we could annotate those differentially abundant clusters as having indeed to do with differentiation and cell type. Cell cycle. Okay, so in the final part of my talk, how much time do I have? Because we started a bit early. Okay. So everything I've spoken so far about has been on the standard version of cell painting, which was developed about 10 years ago, primarily as a screening assay for cancer studies. So it's been used a lot in the context of drug screens and kind of looking for cancer drugs effectively. So that means it was intentionally developed to be super generic so that it could be applied to any kind of cell. It would just capture all the basic organelles and so on. So it was intentionally generic. But of course, not all cells are round. Particularly for us, neurons are certainly not round. And the same is true for other cell types as well. And recently, adapted versions of cell painting have started appearing where the groups have started to tailor the assay more for particular cell types. So in these couple of examples here, the first one is tailored towards profiling lipocytes, and in the second one, it's about T cells, or tailoring it to profile T cell activation, basically. So we got intrigued. We started to think if we could do the same for neuronal cells. So we've been playing around with some alternative cell painting dye panels for a while now, swapping out individual dyes with others, combining them in different ways, just to see if we can improve the assay, if we can learn more about specifically neuronal cells by doing this. Now, the standard cell painting, based on the previous experiment I talked about, we know that it's not very good at picking out cell types. It can basically separate out a round cell from a not round cell, but that's about the extent of it. So that was certainly one question for us in terms of these alternative dyes. Can we do better in terms of just cell type classification? Not because we necessarily want to somehow replace single-cell transcriptomics. That is by far the most superior way still to identify cell types. But in certain experimental contexts, it can be useful to be able to tell certain cell types at least apart. This is a slightly different differentiation protocol here, a quicker one, effectively. The details don't really matter here, but what does, the one thing that does matter is that in this particular protocol, this is a co-culture system. So the neurons are cultured together with rat astrocytes. This is primarily a technical reason to keep the neurons, which are fragile cells, happy in the dish. Similarly to before, we collected imaging data using both the standard cell painting assay as before, and then 3 different versions of our new panel. This is now more cells, about 500,000 cells, and a little bit more features as well. So about 500 or so non-redundant features. And you can see the UMAP there on the right. If I dive a little bit more into what this UMAP actually represents, we have time point differences. The yellow ones are the round ones, the precursor states from the first time point. And then, well, the blue ones are iPSCs, but they're just a kind of plate control. But you have the neurons then on the right-hand side. And the neurons are now a mix of co-culture wells and of monoculture wells. So we intentionally, plated cells in a way that we had wells where we had only astrocytes, cells where we had only neurons, and then the kind of main ones, which were the co-culture wells. Yeah, this is basically what I just said. And then we wanted to ask, okay, given that we have monocultures and co-cultures, are these new panels helping us in terms of differentiating out cells in the co-culture wells? So we turned to something very simple. We used a random forest model to basically train it on the monocultures and then use it to predict cell types in the co-cultures. And we then compared the outcomes both in the standard cell painting as well as the new. And of course, there's a lot of unassigned cells, but the take-home first observation from this was that panel 3 seemed to do best in terms of predicting neurons. It kind of managed to predict most neurons. Actually, I do realize I skipped over something important here, which is that if here the kind of first coloring here is based on the time point, so the precursor versus neuronal status. The one I skipped over here is the coloring based on the panels. So you can see that in both of these time points, there's a split into two clusters. One cluster contains the standard cell painting assay, and panel 2, they don't seem to differentiate very much. So basically panel 2 here doesn't seem to give us very much new information, whereas panels 1 and 3 are clearly distinct. So they are definitely adding something to this story. So in line with that, really, it seems that panels 1 and 3 are in some ways, well, one is predicting more astrocytes, one is predicting more neurons. So we don't quite know what to make of that just yet. However, you can then do this in a more detailed way. We had multiple cell lines in this experiment, for instance, and you can then start comparing the kind of like-to-like comparisons. So basically the correlation of predicted astrocytes to the real astrocytes, predicted neurons to real neurons, and so on. And of course, you would expect here the like-to-like comparisons, which are up there, to be high, and then the cross-cell type correlations to be low. And this is indeed the case. And again, panels 1 and 3 seem to be behaving best with this respect, having the highest like-to-like correlations and the lowest cross-cell type correlations. So this is very much work in progress, but this to us basically tells that if you start swapping out swapping in dyes that are more informative for neurons, you might be able to improve your ability to predict different cell types from your images. Now, as mentioned, of course, this is not our ultimate goal. If we wanted to just characterize cell types, we would probably still use single-cell transcriptomics. But of course, if you can separate out cell types based on the images, this also means that your assay is capturing something really essential about the neuronal cells, which of course then can be informative of disease. So just to wrap up what I've spoken about today, we've been experimenting with cell painting as a new assay to add cell-level phenotypes into the omics mix in single cells. And it seems that it really can capture cell morphology and other kinds of cell-level phenotypes, such as mitochondrial function, which are relevant to neuronal models and disease processes. Cell painting and single-cell RNA-seq are highly complementary. Again, I wouldn't leave single-cell RNA-seq out of this just because we can do cell painting. That's a question I sometimes get, that it's so much cheaper, why would you not just do cell painting? I think they both capture slightly distinct things about the cells. And this kind of joint profiling approach can help us interpret genetic effects in the context of disease models. Of course, when the technology inches forward a little bit still and we can actually get these two measurements from the very same cells, then we don't have to bother with these types of predictive models. Then we actually know the two measurements that are coming from the same cell, but we're not quite there yet in terms of technology. And then finally, that cell type specific adaptations of morphological profiling are likely going to be useful in more specialized cell types, such as brain cells. So yeah, that's it. Thank you very much.
Thank you very much, Helena. Questions?
I see that for dimensionality reduction, you mainly rely on UMAPs and then clustering. You did not mention too much of the algorithm that you used, but then I was wondering, have you played a little bit around the use of modern deep learning methods where they use embedded dimensions as a dimensionality reduction strategy, which, given the nature of the cell painting, where you have totally different data distributions like pixel intensity or cell size object, they can be more suited for this kind of task. Thank you.
Yes. So what was the first question about clustering methods in general? Yeah. So there's definitely a lot lot of choice in terms of methods. It's not really so established as perhaps you would want yet. So we have played around with different tools. For neurons in particular, the segmentation step is hard. It's not easy to segment out individual neurons with, let's say, long axons or something like that from images. So that's definitely a kind of bottleneck and a caveat in all of this. In terms of more advanced methods, certainly this has been sort of a first pass analysis, I would say. But certainly, for instance, in the step where you extract features from your images, this can be much more powerful than what we have here. So you can definitely kind of add more features from the existing images you have using these types of methods, and certainly also the data integration space is challenging. So there, I think those kinds of methods and kind of can be useful. Yeah.
Thank you.
Will be useful for sure.
Other questions, Thomas?
Thanks so much for a really interesting talk. Forgive me, I'm indeed not so familiar with cell painting, and I was wondering, what are the features you get from there? How does it look in the table?
So the features are basically derived from. It depends a little bit on which microscope you have in-house. So this is a confocal microscopy-based assay. In our case at FIM, we have a microscope that has 4 lasers. And so we basically have 6 different fluorescent dyes captured with 4 lasers. And so you have basically features that are coming from each individual channel. So for instance, the mitochondrial stain is in one of them. So you get kind of a bunch of mitochondrial features that are about the dye intensity, texture. There's a little bit of spatial information there as well in terms of how close to other cells your cell is and so on. But these kinds of features. We use a tool called Cell Profiler for the low-level image analysis. But again, for these kinds of more advanced machine learning methods, you could then probably extract many more features than this, which then the downside being they're even less interpretable than what we have here.
Thank you. Great talk. Oskari Kulta, PhD student in neuroscience from Tampere. I think it's extremely relevant what you're doing, like correlating these different levels of data. And we're, for example, now trying to correlate electrophysiology with sequencing data, and also interested in building also towards those image analysis into that pipeline as well. And that is what we are seeing in the literature actually now popping up, especially in these diseased models of in vitro neuronal diseases. But I'm just thinking, I saw your differentiation protocol where you had the rat astrocytes. I know it's really established protocol, many people are using it. And are you planning to get rid of the rat astrocytes at some point? I know it's difficult, but do you have some kind of a pipeline in mind or plan?
Yes. So I'm not a big fan of these rat astrocyte co-cultures either. I mean, if the shift is towards human models, then you don't really want rat cells in your culture, right? But we have tweaked the protocol such that our neurons do survive as monocultures. So we will probably use that approach going forward. The downside being, of course, that the cells are then slightly less happy than they are with the kind of feeder cells almost. So that's a bit of a trade-off. The first protocol that I was talking about doesn't have this, so it's a kind of traditional morphogen-based approach where you put the cells on a path and they just develop into what they're supposed to develop. So that has advantages in terms of that. It doesn't require rat cells, but then it has other challenges. So it's about of picking your evil.
Thank you. Other questions? Aha, there is one or two. Did you try cell painting for solid issues?
Solid issues. We haven't. I think it is applicable to tissues as well. Many use it on organoids, so kind of 3D imaging. So that is possible.
And the second one, neurons are a system where probably more than elsewhere the heterogeneity rather than just the mean of the signal matters. Can you share your view on this?
Yes, so this is kind of related to your question about the protocols. There's roughly speaking 2 ways to make neurons in the dish. One is that you push them super aggressively towards a certain particular fate, in which case you probably get those cells, but the cells it's done in a very unnatural way. And the second way is that you do it in a more gentle way, in which you put them on a path and they kind of happily become a certain kind of neuron. But then, of course, in the second route, which is in many ways better, then they have also more space and freedom to misbehave and make all kinds of random cells and so on. So heterogeneity is definitely both a feature and an issue. Of course, we want heterogeneity. That's what the brain looks like. There's definitely lots of different kinds of neurons, different kinds of progenitor populations, and you want them all there. So in some ways, you don't also want to skip over those developmental stages too quickly. So, yeah.
Thank you very much. I think it is time to move on to the next speaker. I know there were some more questions, but I think we can continue all the discussions during the coffee break. Before we, so maybe once more, thanks for Helena. And before we